So on yesterday, we were, I think this is where we left off. This is the sheet I was writing on for you guys. So I was trying to explain how the genes that you colored yesterday and what we talked about yesterday with the fly and the mouse, how that regulates development. So we, just to bring us back all here together, that we had this gene called a homeotic gene that helps to regulate development. And then within that gene, there's a section, a small segment of bases in the DNA. And what happens is when we transcribe this gene into mRNA, it transcribes the whole message from the gene from beginning to end, including that homeobox region. So then what happens is when we translate that, and so the mRNA goes out of the nucleus, goes to the ribosome, and so on, uses the mRNA and reads it in groups of 3, the codons, and brings the amino acids, and we hook together the amino acids, and then that polypeptide folds on itself to make your finished protein, so that you get a 3-dimensional protein. And so that's what I'm going to attempt to draw here. So let's say here we have this 3-dimensional protein here, and we have curves and so on. Why? Because we have interactions between our groups, right? And that's what stabilizes and gives a protein its structure. But what happens here is, if this is the protein chain that's made from this mRNA, the part of the mRNA, the homeobox part, so I'm going to use the same color here, shows up and is part of the actual region here. So this right here on the protein is called a homeodomain. So this is just part of the protein. And then it's our finished protein. So what happens is, what does this protein do? So once it's in the right shape, what it does is it acts as a transcription factor. Turning on other genes involved in development. So what does it mean it acts as a transcription factor? Transcription factors bind to the promoter region of other genes. And so let's say you have, now here's a piece of DNA. So this is DNA, let's say, from another chromosome. So it's not the same DNA here, we're not talking about the same gene. So the DNA from another chromosome. And in that DNA, you have a gene involved in development. So we have to turn that gene on. So how does that gene get turned on? You have to have transcription factors that bind to the promoter region. So there's the promoter region for that gene. So this gene currently is turned off. So what happens is, these master control genes called homeotic genes contain this homeobox, and they make this transcription factor. What that transcription factor does is bind to the promoter region here. So that's why it means that, so I'm going to put here. So this goes and binds here, and so that's that protein. But what I want to do the color coding here with the domain, the homeodomain. Look at where this is at. It's the part that helps bind to the DNA. So that homeodomain allows and helps that transcription factor to bind to the promoter region, which then other transcription factors will come and bind. So these are other transcription factors. And as a result of these transcription factors binding to this gene, what can come bond and start transcription? RNA polymerase. And so now RNA polymerase can come. That's your RNA polymerase. So the RNA polymerase comes and binds. And now that can start to move down and transcribe that gene and help with development. And so, how this relates to what we looked at yesterday is that, let me put the picture up here. These genes right here, this is like one of the genes that we looked at in the family of genes yesterday. So let's see, let's wait till this comes up here. So this picture here, it's kind of like the picture that you colored yesterday with the fly and the mouse. And so remember we talked yesterday that these genes are found, genes are found in the fly and the mouse, and we color coded them, and we noticed that by color coding them that they correspond to the same type of region in the fly and the mouse. And so how does this relate to that picture that I just looked at? So like that we just drew. So like this little purple segment here for the fly. What that means is that would be like the gene at the top of the picture we just drew. So there's a homeotic gene and there's a part that's the homeobox. In this part of the fly, in the cells on this end of the fly, this gene is turned on. This is a master control gene. Why? Because if this gene is turned on, it produces transcription factors, which we just drew. So it produces a transcription factor that can then go on and turn on other genes in the head region of the fly to help it develop into the head region. That master control gene is different than the orange one on this side here. This orange gene also is a master control gene, like the top of the picture we just looked at. It has that homeobox mark. It codes for making another kind of transcription factor that turns on other genes that make the back side of it develop into the back side of the mouse. And because these genes are different than these genes and make different transcription factors, you have different than genes turned on here than you do here, and therefore the front and the back and every other segment in between develops differently because you're turning on different genes. Does that make sense? Okay. All right, and so those are called homeotic genes. So this I think is where we left off in our fill in the blank note. So is that right? All right. So homeobox is a DNA sequence found within genes that are involved in the regulation of patterns of development. So that's what I tried to draw there. So when it says found within genes, so remember this is the homeotic gene, and the homeobox was just a section of that, and this was the whole gene. So that's the drawing that we just looked at. Genes that have a homeobox are called homeobox genes and form homeobox gene family. No?
Yeah,
what?
lunch. Yeah. Is the door's locked?
Do you need another blanket?
No, I can write it down.
The most studied and most conserved group of homeodomain proteins are called the Hox genes, which control segment patterning during development. So these Hox genes are this is an example. This is like those. They're a collection of genes that the same ones like that we color coded yesterday. Selection of genes that, when turned on, turn on different genes in different parts of the body that ultimately end up in segmentation. And so how we get the segmented. But there are other kinds of homeodomain proteins. So not all of them are involved in segmentation. The Hox ones are happen to be involved in segmentation. So this kind of talked about this yesterday. Many distantly related eukaryotes, such as plants and yeast, also have these same genes, the same collection. of genes called the Hox genes. They're regulatory sequences, which means that they're able to turn on or off genes. Such similarities indicate that the Homeobox sequence is very useful in development and has arose probably very early on and has been conserved and found in organisms for hundreds of millions of years. All right, so because lots of different organisms have them. Not all of these Hox genes are homeotic genes, which means that they have a homeobox. Not all of them control body parts, however, most are involved in development. And so we can think of Hox genes as the segments, the controlled segments in us. So this research has revealed that these genes, that these homeobox encoded regions, is part of the protein that functions as a transcription regulator, turning on and off transcription. The shape of the encoded region allows it to bind to any DNA segment, but by itself, it cannot select a specific sequence. The variable regions within the whole protein allow it to interact with other transcription factors and enhancers within the DNA. And in this way, the Homeobox genes work to switch certain developmental genes on or off. So this second bullet, what that's talking about, going back to our picture, is that, so the shape of the encoded region for the Homeodomain, allows, move that, this part here, allows it to bind to the DNA, but then that in itself isn't going to turn the gene on. So the whole protein is important because that helps the other transcription factors to bind, which then eventually leads to RNA polymerase to bind. So the whole protein is important, not just the Homeodomain. All right, and so that's how they turn genes on and off. All right, and in regulating transcription and translation. Alrighty, and that is chapter 21. Okay, so I want to spend a minute before I'm going to pass back your test. I got everybody to take the test yesterday. Before I do that, I want to talk a minute about kind of this unit that we're doing. So kind of give you an idea, because it's kind of can be confusing because we're taking little bits from different chapters, and I just want to pull it all together for you to make sure that you see the big picture. And so I just want to spend a minute and talk about that. So let's look at what we've already talked about with chapter where we are right now at this moment. So since our last test, we ended up with our last test, the last thing we talked about was protein synthesis. So then we've done this little mini unit with parts of different chapters that have to do with gene expression. So, and gene expression is different in prokaryotes versus eukaryotes. So we talked about chapter 18.4. So remember that was with the operon, so the lac and the trip operon, and how bacteria turn on and off certain genes. So then we compared that to eukaryotes. And that's where we looked at parts of chapter 19. So chapter 19, we did certain sections of that. So in that chart, we reviewed and went over as a review that chart that shows you like DNA methylation and then how we control transcription with enhancers and the activators and so on. And so that was all chapter 19. And then the last few days we were relating that to development and how genes are turned on and off very early on in development with the zygote and the embryo. So that was chapter 21. So notice here we're doing segments of chapters, so it's not full chapters. And so what we're going to do next is do a little bit, this is we're kind of ending about DNA and genetics and that kind of stuff, ending the whole kind of topic on that. And so what we're going to do is take now everything that we've learned about DNA, genetics, gene expression and so on, and apply it to how we as humans manipulate that. So we make what are called, for instance, transgenic organisms, where we can take a DNA and genes from one type of organism and put it into another type of organism and have it do certain things that we want it to do. So we're going to apply some of the things that we've learned. And so that's the last little bit of this kind of molecular kind of genetic thing. So we're going to do a little bit and talk about an application of how we manipulate genes and look at some DNA technology. I'm going to pick and choose certain ones. This topic is really a huge thing. We could spend probably a whole semester on DNA technology. But I'm going to pick out certain things that you need to know for the AP test. So in order to understand this, we have some background information that we need to know. And so that's what you picked up today. We're going to talk a little bit about bacteria and viruses. So the first 18.1 is about viruses and 18.3 is about bacteria. We've already talked a little bit about viruses and a little bit about bacteria. I'm just going to give you a little bit more information that you need to know in order to understand this DNA technology. Now, you wouldn't think probably that bacteria and viruses have much to do with DNA technology, but what we do to manipulate genes is use information that we know about these guys and use things that are found in nature to our advantage. And so it helps to have some background information so that when we get to the actual technology part, we're just going to do a few parts of chapter 20. It's not the whole chapter. That will apply this, and this is where we're gonna do the putting a jellyfish gene into the bacteria and make it glow. All right, so that's a 3-day lab. So let me give you just an idea. So today we just finished 21. I'll give you your test back. Most likely the other hours I didn't get to that packet with chapter 18. So tomorrow we'll talk about bacteria and viruses, and then we're going to start getting into chapter 20. And I'm going to teach you some things. I have a couple of simulations, like paper type of things, so you can visualize what's going to go on. And then next week we're going to have 3 days that we're going to do the lab. Early next week, I'm thinking Monday through Wednesday. And then we're going to have a test by the end of next week. And this is the stuff that it's on. Okay? So I just want to give you kind of an idea of how everything fits together and the plan. Okay? Alrighty. Alright, with that said, I am going to pass back your tests.